Right. Hello and welcome. This is my part of BIOM24. We are going to be spending 3 hours together talking about bacterial secretion systems, which is something that I'm really excited about because I used to work on type 3 secretion systems, as you probably know by now. So I'll spend a lot of time talking about that and also other contact-associated type 3 bacterial secretion systems, and some time talking about the others. So today I'll just try and give you an idea of how amazing machines these secretion systems are, because they truly are. I mean, you know, if you we'll compare them to some other man-made machines and things, and you will just see how amazing they are and how simple yet beautifully elegant work. And then in the next lecture we'll spend some time talking about how and why we study these secretion systems and the sort of methods that people use in the lab to discover new things about secretion systems. And the third lecture I've titled The Weird and Wonderfuls. We'll talk about atypical secretion systems and unconventional uses of normal protein secretion systems, which will make probably more sense once we get to that part. But it's largely about other ways that bacteria use to secrete things, and also how we can engineer secretion systems to use them for our purpose, be it biomedical applications or biotechnological applications. Because I think they have a really huge potential for us to engineer them in a way that they become useful to us, because there are such amazing ways to interface bacteria with their environment, or even bacteria with host organisms. I've included some papers here. As usually, you don't have to read them, but there are just some recommendations for further reading, and you will see that I've sort of tried to incorporate the headings of different papers that I will be talking about and touching a little bit upon, but you can read for further information, and I've embedded them as we go along. So this is a slide that just gives you an overview of all the conventional protein secretion apparatuses that we have discovered so far. And when I say discovered, that is largely an overstatement, because we have, for some of them, only just begun to understand that they are there, and we know nearly nothing about the way they work. Whereas for others, we have quite a good idea already how they work and how they look like by structural studies. So I will show you some of the structures and what we know about how that explains how they work, and then talk about the methods in the next lecture that people use to sort of explore the structures of these sometimes ginormous structures. So largely we have type 1, 2, 5, 3, 4, 6, and 7. They're just grouped by the way they work rather than the numbering, because that really doesn't mean a lot. So the type 1 secretion apparatus on the far left, it has an ATPase subunit in the inner membrane. So we always have inner membrane here, outer membrane here, and then sometimes there is the third layer which should just depict a host membrane, plasma membrane of a host cell, if that is relevant to that secretion system. This one here is a bit different because it's mycobacterial membrane, because this particular secretion system has been found in mycobacteria, and that's why the membrane looks different here. But all these are largely gram-negative organisms, and in the inner membrane there is an ATPase subunit for the type 1, and then it translocates its substrate out of the outer membrane, and these are generally then secreted into the extracellular medium. Whereas type 2 and type 5 rely on some of the general transport mechanisms that bacteria use to transfer proteins into the periplasm. So you probably know that there are 2 general secretion pathways: the Sec-YG channel, which translocates directly from proteins emerging from the ribosome, freshly synthesized proteins, and translocates them into the periplasm, and it translocates them as a nascent chain, so they are at that point unfolded and unraveled, and they're not discretely folded as proteins as they are being translocated. Whereas the TAT pathway here is a pathway that recognizes slightly different signal peptides on proteins, and that is dedicated to proteins that need to be translocated in the partially or completely folded form. Okay, so that's the difference between SEC, which locates unraveled or unfolded proteins, and TAT, which translocates folded proteins. The reason being that proteins that go through the TAT pathway, as you've probably heard, they usually require cofactors that are only available in the cytoplasm in order to assume the native structure. So that's the reason why they have to be transported in a folded state. So there might be cofactors like heme or iron sulfur clusters that they need to incorporate into the structure in order to function, and these are only available in the cytoplasm, so they are folded there, including the cofactors, and then translocated through TAT. And type 2 and type 5, depending on the protein substrate, can use either one of these pathways to get the protein into the periplasm, and then from there on, there are discrete pathways that are either type 2 or type 5 pathways that secrete them into the extracellular space. And we'll talk about it more later on, but these pathways can transport not just proteins, but also other substrate. They can transport small molecules, sometimes they can transport DNA, they can transport polysaccharides, so there's quite a diverse range of substrate that can be transported through these pathways. And then we have types 3, 4, and 6, which are all contact-dependent secretion systems. So all these are used by bacteria to transfer substrate from their own cell into a host cell. And in the case of type 3 secretion, the host cell is a eukaryotic cell. You know, it can be a human cell, an animal cell, a plant cell. The same is true for type 4 secretion. However, for type 6 secretion, it can be either a eukaryotic cell or, as we will see later on in the movie, another bacterial cell. Okay? But all these three are contact-dependent translocation systems which transfer substrate from one cell into the other, which makes them quite distinct from the other secretion systems. And then we have the most recently discovered one, which is type 7, which has been found in mycobacteria, but has more recently also been found in other gram-positive organisms. And there's really not much I can tell you about it at that stage, because we're only just beginning to understand what the components are of the secretion systems, and we really don't know much about what sort of role or function they have in the context of bacteria and their lifestyle, but I will tell you a bit more about that later on. Okay, so this is just again a reminder of SEC and TAT translocation pathways. Yeah, so what I've just said. So basically for SEC secretion, there are at least two ways that the translocation can take place, either as a nascent chain directly from the ribosome that gets delivered to the SEC-YG by the signal recognition particle in blue here, or they can be SEC-B dependent substrates. So that takes place co- or post-translational. So it could have already emerged from the ribosome, then SEC-B takes it up, it's a chaperone, so it leaves it in its unfolded state and then delivers it to SEC-YG. And it's an ATP-dependent pathway, so ATP drives the process of translocation through SEC-YG, and then emerges in the periplasm. And like I said, TAT pathway has a different signal peptide and transports folded proteins into the periplasm. Okay, so type 1 secretion systems. You'll probably see on some of the slides I have put keywords down at the bottom, so as we go along, I will try and explain where this is going. If you are clever, you probably should make arrows or label some of the components with some of the words. I just put them there to remind myself mainly to what to say to you and which terms to explain to you, but you can use it to label some of the components. Right, so type 1 secretion systems, they can transport incredibly big proteins sometimes, right? So what's depicted here is the translocation of an RTX toxin, which occurs in things like Vibrio cholera. We have talked a little bit about that. And it consists of an APC transporter subunit. This is the substrate down here. It's got a secretion system, secretion sequence that's recognized by this inner membrane component. And then there is a membrane fusion protein, MFP, which is this green part here, and then an outer membrane poring component, which is shown with the structural depiction here. And so the protein directly is translocated from the cytoplasm into the extracellular space without emerging in the periplasm. And in the extracellular space, where there's calcium, it uses that calcium binding as a driving force to fold the protein back up. And once the substrate has completely emerged from the channel, the apparatus then resets itself. So the components usually only associate when the substrate loaded into the ABC transporter component. And then it docks with the outer membrane component using that membrane fusion protein, and then translocates and is competent to translocate the substrate through. So I think type 1 secretion systems are a really nice example of where structural discovery from individual components has sort of led the way in understanding how the system works. It's interesting because obviously this is a huge machine that transects the whole cell wall in a membrane all the way through the outer membrane. But we have discovered some of the structural subunits independently from each other. So we have got a structure, we had the structure first of the inner membrane component, and that structure was a dimer, okay? So 2 protein subunits forming that inner membrane complex. And then later on, people characterized the outer membrane component of the system, which is shown in red here, and they found it's a trimer, which was sort of puzzling because trimer and the dimer don't really interface very well. So people kind of were puzzled about that finding and said, is the structure just an artifact or what is going on? And then it sort of came up the idea that there must be a subunit in the periplasm, which is this membrane fusion protein, the blue part here, which links these 2 subunits together. And the beauty of it is, it has a very clever way of doing that, is that this is a hexamer, okay? And hexamer being just the only really logical explanation how you'd link a dimer to a trimer symmetry, right? So I've sort of made this, I said I would kind of bring up examples of machines. I kind of like these comparisons. So this is not really biomimicry, but it's sort of humans trying to come up with similar solutions to problems than biology does, right? So the problem here being a symmetry break, right? How do you translate the symmetry of a dimer into a trimer? Well, nature uses membrane fusion proteins that have hexameric symmetry. This is a gearbox from a car. Okay, did someone recognize that? So we sort of have the same problem where, you know, we translate differential speeds from one component of a moving car to another one. And Audi, this is an Audi gearbox, which is sort of the prototypical gearbox. How many components does it have? You guess. 3,100 components, 3,100 components, okay? This here has a trimer, hexamer, and a dimer, and it does the exact same job, really, you know, it translates symmetry. This is just a beautiful example of how something like a protein complex that has evolved over time in biology comes up with a much more elegant and beautiful solution to the same problem that we have struggled. You know, and these rarely break, and you can switch over the parts as it is moving and translocating substrates. Yeah? If you have wear and tear, which you have in protein complexes just as much as in cars, be it in flagella, be it in protein secretion systems, the bacteria can exchange broken parts for a new one and just replace them. And single molecule studies and single cell studies have showed us with things like flagella that while they're doing this, the flagella are still working. Okay, they may decrease the speed, but they're still working. Okay, it's like changing the wheel on a car that's moving on a highway, right? It's quite an amazing feat to do that. So that's why I like protein secretion systems. There are just some more type 1 secretion systems. That's to show you that most of them are associated with drug efflux, and you've probably come across them, and you should revise. When you revise, you should refer back to Andrew Levering's lectures, because he talks about, you know, antibiotics and resistance and drug efflux a lot as well. And I'm sure he's got a very much more interesting perspective on that than I have. But that's just to say lots of different organisms have them. And mutations in drug efflux pumps, so type 1 secretion systems, are one of the most commonly found mutations in drug-resistant organisms that are independent of the substrate, or largely independent of the substrate. And they're just, you know, sometimes individual amino acid changes in the sequence. So you probably heard a lot about plasmid-based resistance mechanisms and horizontal gene transfer and things like that. Well, that's a very easy solution for a bacterium to become quite intrinsically highly drug resistant is by doing individual base pair changes and amino acid changes in type 1 secretion systems. Okay. This is just a nice paper I found about different drug efflux inhibitors. So one of the ideas that people have come up with is because they found all these resistant bacterial strains that pretty much share different mutations that makes the drug efflux systems more efficient. So they pump out the drug a lot more than wild type strains would. So one approach to drug resistance then is to screen for small molecules that we can use as inhibitors of drug efflux pumps, because if we inhibit or block the pump, the bacteria can't get rid of the antibiotic anymore, and that would be a good solution to give the patient alongside antibiotics to avoid drug resistance problems. And this is just a screening approach depicted here, and if you are more interested in it, you can just read the paper. It's basically showing a wild type E. coli, and the assay uses a fluorogenic substrate, so it's got a substrate here that becomes split into two halves if it's inside the bacterial cytoplasm, and if it gets split or broken in the middle in the cytoplasm, it becomes fluorescent green. Okay. And you can see here different components of the pump. So you have the outer membrane protein, Tol-C, which is one of the type 1 secretion system pumps. And then you have ACRB, which is the inner membrane component of that particular pump. And there are other pumps that can sort of partially complement ACRB. So if you delete ACRB, the inner membrane component, the bacteria take up the substrates, they cleave it, it becomes fluorescent green. But you also have other pump component in the inner membrane that can partially complement the loss of ACRB, so that the pumps sort of not very well, but still pumps out the fluorescently green substrate. So you get bacteria that are green, but also environment that is green, which is shown here. However, if you knock out the Tol-C, so the outer membrane component of the pump, it can't be replaced as easily. So the bacteria take up the substrate, they split it in half, it gets fluorescent green, and then the substrate gets stuck inside, which is what you want. So with some inhibitors, if they are working, they will mimic the effect of loss of Tol-C, because they block the function of Tol-C, so the substrate will be split and get stuck inside. So in that assay, as they are screening different small molecule inhibitors, they are essentially looking for a compound that will render the bacteria green without having the outside green as well. Okay, so they are looking for a substrate that mimics this state here, and this is just the way they have set it up, is by doing microfluidics, where you have several parallel channels that you can load with with the bacteria, but different inhibitor components that they are screening, and they are essentially looking for the level of fluorescence in the bacteria. And if they have identified a component that is a good inhibitor, you will see that the bacteria will light up green, just as if they would do if they lost Tol-C. Does that make sense? I see a lot of puzzled faces, I think. Yeah, do you have any questions? You just stop me and ask, please. Okay. If not, we're moving on to type 2 secretion systems. I should probably show you that picture first, because it's sort of, I'm a quite visual person, so with things that are quite complicated, it sometimes helps me to visualize them with very, simple things that we use all the time. So type 2 secretion systems work very much like an elevator, okay? Or escalator, we call it, where the stairs are emerging from the bottom and move up, and then they're kind of recycled in a way. So type 2 secretion systems work pretty much the same way. They have an inner membrane component, and then they have a pseudopillars which can emerge and from the inner membrane component and dock with the outer membrane component here, and sort of integrate subunits of GSPG here, the pink one. They can sort of be taken up by the inner membrane component, and then in an ATP dependent manner be integrated to form a structure here that essentially pushes a substrate, which is shown in yellow here. This is the substrate in our case. Push the substrate out through the outer membrane component until it emerges into the extracellular space. And then that sort of the pilus subunits are disassembled again, and the system resets itself and gets ready to load another substrate. So yeah, inner membrane component, ATP dependent, has this platform that it can load a substrate onto and then extend subunits, polymerized subunits, to extend through the outer membrane component and basically push the protein substrate or substrate out like a plug from a bottle, right? Yeah, so that's very cool as well. And we have found something very similar, which is an escalate. Yes?
So when the pilus goes through the outer membrane, do the monomer units break down and then go back in? Or?
So it senses the presence of that protein that it has loaded, the yellow bit here. And it keeps assembling from the bottom up to extend all the way to the outer membrane. And what it senses then is the loss of that substrate and the emergence of the substrate and the release of the substrate into the extracellular space. And once that has happened and it's lost the contact with the substrate, then it will disassemble again. Right. Type 5 secretion systems are the next one. It's the other one that is dependent on an inner membrane component that's shared with just common secretion substrates. So it takes either sec, YG, or TAT, and then transports the substrate into the periplasm. It's cleaved, so the signal peptide after translocation is cleaved by an inner membrane protease. And then it docks to an outer membrane component, which is a beta barrel. So this here and this here is outer membrane components, and that one is called OMP85. So it's an outer membrane porin, and it recognizes the sort of C-terminal part of the protein, which is protected by chaperones, so it doesn't fold in the periplasm already, okay? And it docks onto OMP85. And that makes, so it doesn't go through OMP85, okay? But rather OMP85 then starts putting the N-terminus of the protein into the outer membrane. And when it happens, it starts assembling into a beta barrel as well. Okay, so beta barrel assembles another protein as a beta barrel also next to it in the outer membrane. And then the protein essentially goes through itself. So it takes the N-terminus, inserts it in the outer membrane, forms a beta barrel there, and then threads the rest of the protein through to itself. And as it emerges on the extracellular space, it loses the chaperones in the periplasm, and then it starts to fold, okay? And it's actually quite a common system that is used. It's an autotransporter system, right? And it's a common way that bacteria put stuff on their own membrane that remains there. So a lot of the adhesins that we know, or some of the clustering components that bacteria use to form microcolonies, are type 5 substrate and are autotransported proteins. Not all of the substrates stay there. So here's one like that. It's intimin. It's what E. coli uses as an adhesin to adhere to host cells, right? And that's an autotransporter protein. But not all of them stay on the outer membrane. Some of them can cleave themselves and actually be released from the bacterial membrane. And some of the toxins that we know do that. There are two different versions of these type 5 secreted autotransporter proteins. Some are monomeric and some are trimeric components. So the trimeric ones, which is one of them is shown here, is actually such that they have a trimeric coil as the C-terminus of the protein, which is the part that's extracellular, and each one of these three monomers contributes a third of the beta barrel that forms in the outer membrane. Okay, so this barrel is formed by all three proteins subunits together. This is just showing the same, so you can have monomeric where the whole protein forms a barrel, or trimeric where each monomer forms a third of a barrel, or you can have a barrel that then cleaves the C-terminus of the protein and releases it. This is just one really nice paper that I found in PNAS, which uses a protein that's called pertactin, which forms this awesome fold here. and using that showed that the driving force, because obviously you're faced with a problem, the ones that are coupled with the inner membrane directly, those protein systems usually use ATP hydrolysis as a driving force to drive translocation, right? Whereas the type 5 one is one that has a brake, right? You put stuff in the periplasm using SEC or TAP, which are ATP dependent, but then it's there, right? And how do you get the protein to translocate through the outer membrane? What's the force? There's no ATP in the periplasm. So what happens is that the fold of the protein, as it emerges and it folds, the folding of the protein actually gives the energy that drives translocation through the pore, okay? And this was shown here. So over time, the protein emerges at the extracellular space, and you can detect it by degradation. So it's protease sensitive once it emerges from the channel, and they use a protease K assay to show how quickly that proceeds over time. Okay. So in the beginning, you have intact protein because nothing has emerged yet, and the protease can't cleave anything. But then over time, as the protein emerges, it becomes protease sensitive, and you can detect the time that it takes the protein to translocate through the outer membrane. And this is the same thing. It basically shows how the protein is folding over time as it emerges from the channel. Yeah, and pertactin is also one of the proteins that are used to bind host cells. So this is the RGD loop, which is a motif that is used by different bacterial proteins to bind host cell integrins. This is just another example of an autotransporter. I said some of them are used as bacterial recognition. Rather than sticking to the host cell, to use these proteins to stick to other bacteria. So one of those is TBA, and this is just to show a neat assay for biofilm formation. So the protein is recognizing similar cells on the surface by TBA interactions. So they have made different strains where the cells are either red or green, and they are the same cells, though, same type of cells. They've mixed them together, and they have shown that usually the cells clump together, but in the knockout they don't clump as well. So there are different. You can either do the mixing assay to show that, or you can do a confocal reconstruction of the biofilm that they form, and you see that in the mutant the biofilm isn't formed very well, whereas in the wild type bacteria they form a very nice fluorescent biofilm. And this is just the top view, and that's the side view. And that's another assay to look for clumping. So that's just bacteria shown by microscopy in phase contrast. And these are free and these clump. So that's a mutant. And the ones that clump, they sediment in a tube towards the bottom of the tube, so you can measure degrees in OD. They're just different biofilm assays. So moving on to the ones that were really cool, which is type 3, 4, and 6. So we've talked a little bit about type 3 secretion systems before, and I just want to say, feel free when it comes to your exams to use any knowledge you have about type 3 secretion systems from any lectures ever that you have heard or anything you've read at all. We've talked about it a bit. It's like a hypothermic needle, which is another device that we have built, which is essentially quite close to the biology of type 3 secretion systems. And it's contact dependent, so it's got an inner membrane ring, it spans the whole periplasm, it's got an outer membrane component, and it forms a needle complex, which at its tip has a translocon, which can form a pore in the host cell plasma membrane, and then translocate proteins from the bacterial cytoplasm directly all the way through to the host cell cytoplasm without ever emerging, right? We've had this debate whether or not it emerges in that, and it probably does not. It's also ATP dependent, and in the bacterial cytoplasm there are chaperones that protect the protein from being prematurely folded, because it has to thread through probably in its nascent state, so unfolded. Yeah.
Oh, sorry, but just this the needle
Mm-hmm.
in a T3 secretion system. Did it actually change the shape of the outer cell membrane of the host?
Oh, I see. No, probably not. It's a bad depiction. What happens though sometimes is once it secretes proteins, effector proteins in, quite a common role of some of the effectors is to remodel the actin cytoskeleton in the host, which is what's depicted here. Okay, so it's actually the effector proteins that are translocated rather than the translocon itself. But some of the bacteria, we've talked about things like Shigella and so on, their effect is largely that they encourage the host cell to make actin protrusions, which form essentially a cup that can take up the bacteria. Yersinia likes to do that as well. It causes the actin cytoskeleton to emerge, sort of like it's depicted here, and make a cup that then draws in the bacteria. It's things that like to invade host cells. They like to have effectors like that. This just to show that we have a kind of good impression of some of the individual components of type 3 secretion systems, and these are just individual components that we have solved by X-ray crystallography, and then we use electron microscopy, we'll talk about that more next time, to sort of assemble all the individual crystal structures in what we think the actual machinery will look like. And these are all the subunits and components that we have solved so far. So we don't really know as yet very well about the outer membrane structure in very much detail. But what we do know is that the way they work, they have actually evolved from flagella progenitor, if you want. So a lot of the way of how the inner membrane components work and how the needle apparatus emerges comes from homology to flagella apparatus. And I've got this movie that I wanted to play you just because it beautifully shows how it works. Because the assembly of the components is conserved as flagellum as well. I don't think it has a narrative. So that's the inner membrane component. And then as that is assembled, it's then competent to secrete the components that are necessary to build the periplasmic and outer membrane part. And then once that part is assembled, it becomes competent to secrete subunits that form the needle complex. In that case, it forms the flagella components, but with type 3 secretions, it's just like that. Once the outer membrane components made, then it secretes subunits that form the needle complex. And once the needle is finished assembling, there's a protein that is essentially like a ruler that regulates the length of the needle. Then it translocates the translocon, which forms the pore, and once that is there, you can sense the host contact, and then it becomes competent to secrete effector proteins. So it's got this, it's a secretion machinery that's very complex, and so the process of assembly is very complex, because if it screws up any one of these secretion sequences, obviously it's blocking itself, right? So if it starts secreting effector proteins before it has formed the outer membrane component, it would be just a broken machine. But it's very good at sensing when it's finished the individual stages of building itself, and then becomes competent to secrete the next set of substrates. And like I said, this can keep moving as it, for example, exchanges individual subunits in its inner membrane ring without ever stopping. It's quite amazing. Okay. Back to here. Okay, great. Just find that slide again. Right, this is just a summary of what I told you, that there are early, middle and late stages where it assembles the critical components, and once it's finished that building stage, it will move on to the next stage and start secreting the next set of substrates. Okay? And this is just to show a chaperone as it wraps around the substrate, an effector protein, and then lets go only once it's docked to the inner membrane component. The chaperone comes off as it's read through, so that then the protein never becomes folded until it emerges at the other end. And that pertains to legs. So I said there is a protein that essentially acts like a molecular ruler. So, you know, all these are clustered together on the genome, so people already knew that this protein plays some role in type 3 secretion. But it's sometimes not very trivial to say, is it a structural component, or is it a component that's important for translocation, or is it an effector protein? Sometimes these things are not easy to tell. And with the ruler protein, it was very cool because they tested what the phenotype is when you delete the ruler. And what happened is that the needles, which you can see electron micrographs of here, these are normal needles, okay? And this is what it looks like when you delete the ruler protein, okay? They just formed really long needles all of a sudden. They're like, oh, it must act like a ruler and control the length of the needle and tell the cell when the needle is long enough, okay? So, yeah, if you're interested in that more, you can read that paper. With type 3 effectors and actually a lot of the other effectors that go into host cells, they're incredibly diverse in their activity. And one of my most favorite things to read about and to do when I was a postdoc was to do biochemistry to try and uncover what the proteins do, right? Because we know they are in a way contributing to manipulation of host cell functions, okay? So they mess up the signaling network in host cell in a way that favors infection, okay, that helps the bacteria to establish themselves in the host organism. But if you start reading about what some of these proteins do, I mean, it's just completely mind-boggling, okay? They're targeting nodes in really ancient signaling networks. So if you look at MAP kinases, which are important for proliferation, cell survival, the proteins, they will have diverse functions. So some of them modify proteins in the eukaryotic host in a way that encourages a certain signaling pathway. Some of them will modify them to inhibit a certain signaling pathway. Some will make something like a platform to reassemble a completely different signaling pathway by bringing components together that haven't been together before. They are just complete champions at knowing about eukaryotic signaling mechanisms. So by studying these bacterial proteins, we can actually learn a lot about underlying mechanisms that pertain to eukaryotic signaling mechanisms, because they know how to manipulate it. So if we understand what they do to manipulate them, we know a lot more about how eukaryotic signaling pathways are wired. And these are just some of the conserved themes that come up. If you look at the effectors, like I said, they can either interfere or modify signaling pathway. They can have enzymatic functions. So by studying some of the Vibrio type 3 effector proteins, we've talked about that in the Vibrio lecture, we have uncovered completely novel biochemical modification, right, where there are enzymes that can transfer AMP onto a protein to ampylate it rather than phosphorylate it or acetylate it, deampylate it, right? There have been found bacterial effectors that transfer phosphocholine onto proteins to modify them. So these are completely novel modifications that we haven't known before, but as we look, we find them in the host cell as well, and we're just beginning to understand what purpose they have there, right? And it's a very exciting field. Did I have? No. There are type 4 secretion systems, which are incredibly complicated. I'm not gonna make you, I won't ask you anything about this, okay? So don't worry too much about the assembly. Ideally, what you should know for each of those machines is to roughly draw a scheme that it has an inner membrane component, an outer membrane component, whether or not there is a component in the periplasm, roughly how the mechanism works, okay? I'm not gonna make you draw this or anything else, okay? You should roughly know what type 3 secretion system looks like in a needle and how an autotransporter works roughly, but I'm not gonna ask you a whole lot of detail about it, so don't worry about it too much. It's just to show again that for some of the components we've got good structures already, so we know the ATPase subunit, how that looks, it's a hexamer. We know some of the components of the inner membrane ring, but we know some of the outer membrane component, but we don't really know a whole lot of how the complex works and how these components come together. There has recently been a structure. I'll go probably more over that in the next lecture to tell you, because there have been quite good advances in type 4 secretion system structures recently, so we'll go over that more next time. But it's just to say that they have become very popular because they are found in plant pathogens. So, what do I have that? No. So you can, for example, find Mycobacterium tumefaciens where it causes, it's a plant pathogen, injects effector proteins into plant cells, and it causes these tumors to grow, for example. And it injects, some of the type 4 secretion systems injects DNA rather than proteins into host cells. So they have been used a lot to engineer plant cells, so to make knockouts, for example, in plants. So there have been, by studying bacterial secretion systems, it was at the time quite a basic discovery, right? So yeah, you have this plant pathogen which has this secretion system, but actually it's led to a discovery that's enabled people to engineer plant cells, and it's really driven plant biology as a field. So this is just cutouts of leaves of a plant which have been infected with Agrobacter tumefaciens, and you can see, and they use that as a tool to transform it, and they transport it back onto the plant and then make a mutant that way. And it's also used by disease-causing pathogens, so Legionella pneumophila, for example, uses type 4 secretion systems to invade macrophages and replicate inside the vacuole. So the way that works is actually it causes the macrophage to take it up into a vacuole and then uses type 4 secretion system to inject proteins from the vacuole into the cytoplasm rather than from outside into the host cell. I don't have time to go over that. This is really cool. So some years ago there was the discovery of type 6 secretion systems, which in part mimic very much the structure of a bacteriophage. So that's an EM picture, very old one actually, of a bacteriophage, right? It's got its head, it's got the sheath, it's got sort of tail fibers that it uses to sit down on the bacterial surface. And it's injecting its DNA from, which is packaged into the head through the sheath into the bacterial cell, and that usually integrates into the genome and then does stuff. This is an EM picture of a bacterium that has phage attached to it, which is really cool. And then this is to show what a phage looks like and what type 6 secretion system looks like. So as you can probably see, it's missing the head, obviously, because the substrate comes from the bacterial cytoplasm, but it's got a structure that looks very much like the sheath and the base plate and the spike. So how it works is actually like it's a spring load mechanism. So it loads a substrate and then it disassembles the subunits here to drive the polymerization of the periplasmic component, and then has the spike here which punctures the membrane of another bacterium or the host cell and then shoots the substrate into it. So the force of disassembling this part here drives this assembly of an extension of the type 6 secretion system. I've got a video because it shows it off better than I could have explained.
The bacterial type 6 secretion system is a spring-loaded poison dagger. Certain Vibrio cholera strains, like the one seen here, harbor the type 6 secretion system. By tagging one of the proteins of the system with GFP, we discovered that in live cells, the type 6 secretion appears as an elongated structure that first assembles, then rapidly contracts, and finally disassembles. The cycle can be seen well in these close-up views of individual cells. Whereas assembly occurs over tens of seconds, firing is quick and takes less than 5 milliseconds. We observed up to 5 type 6 secretion structures per cell. In order to obtain higher resolution views of the structures, we also imaged the cells in a cryo-electron microscope. This is a 3D cryotomographic reconstruction of a single cell shown slice by slice. The large tubular structure anchored to the inner membrane is a type 6 secretion apparatus.
So what you can see here is just essentially a slice through a bacterial cell. You see the outer membrane, periplasm, inner membrane, and the cytoplasm. And this is the part that pre-assembles in the cytoplasm, then loads a substrate. And what you'll see is that it depolymerizes, and it polymerizes on top and shoots the substrate out.
As seen here. Our data suggest that the type 6 secretion system is like a spring-loaded dagger. That means a contractile outer sheath surrounds an inner tube. Following some unknown signal, the outer sheath contracts, pushing the inner tube out of the cell. Here is a 3D reconstruction of another Vibrio cell exhibiting an empty sheath in its contracted conformation. Compared to the extended conformation, contracted sheaths were shorter, had a larger diameter, and had a helical surface pattern. Here you can see a central slice through a contracted sheath. After firing, the sheath is disassembled by a AAA ATPase, shown here in orange. A new base plate, inner tube, and sheath then assemble so the cell is ready to fire again.
Okay. No. Yeah, so this is just one example of what type 6 secretion is being used for. It's two different kinds of bacteria, usually bacteria that share the same environmental niche to fight each other, essentially. Okay, so you probably know that there are a lot of mechanisms bacteria can use to kill related bacteria. There are things like colicins, bacteriocins that are toxic to other bacteria. There are contact-dependent inhibition systems, and type 6 secretion systems is one of the more recently discovered mechanisms that bacteria use to fight each other. I don't really have much time left to talk to each other. To talk to that in detail, but there is a really good YouTube video about it as well that explains that paper, which is called Tit for Tat Type 6 Secretion System Counterattack During Bacterial Cell-Cell Interactions. And essentially what it comes down to that you have Vibrio cholera and you have Pseudomonas aeruginosa, okay, and they don't just fight each other all the time. What happens is that one of them assembles type 6 secretion system. They both have type 6 secretion, right? Pseudomonas and cholera. One of them assembles its type 6 secretion system and shoots a substrate into Vibrio cholera, right? And then Vibrio cholera senses that as an aggression and then uses its own type 6 secretion system, which is more powerful apparently because it's got better substrates, and shoots stuff into Pseudomonas, which then carries on to die. And that's called type 6 dueling. And it's a more recent discovery, and they have really good videos where they've labeled Pseudomonas one color and the cholera its other color, and you can see that duel going on. It's really cool. It's like a Western in bacteria. Yeah, and that's just to say there are a lot of different effectors in type 6 secretion, just as there are in type 3 and 4 secretion. And the ones that target other bacterial cells obviously are very different from the ones that they are using to target eukaryotic cells. So rather than manipulating MAP kinase pathways and manipulating immune cell functions and things like that, they target bacterial components. So there are type 6 secretion substrates that target peptidoglycan. Well, that's a very, very common mechanism, very common function of type 6 effector proteins, that they degrade peptidoglycan. Another one being that they target cytoplasm of other bacteria, and it goes all the way into the cytoplasm of the enemy bacteria and degrades its DNA. So it's sort of conserved between bacteriocins and type 6 effectors. The things they target are very much conserved. Okay. Yeah, and that's type 7 secretion. I will just briefly mention it. I mean, there's really not all that much I can say about it, other than it's been found in mycobacteria. It's now been found in other gram-positive bacteria, but it's mainly being studied in mycobacteria still. It seems to target eukaryotic cells. It seems to very much work like other host-targeted secretion systems in that it manipulates host cell function to the bacteria's advantage. But how the components, several components have been identified. Okay, you can knock them out. You see their effect is loss of function. But they are still very much in the process of characterizing which components are structural components, which components are effector proteins, and which of them are both. So they have a really hard time to figure that out, because If you knock a component out and you lose function in that you lose the host effect, it doesn't tell you anything about the way they work in the bacteria cell. So we'll have to rely on future structural studies on them to really figure out what they're doing in the sense of whether or not they form structural components or effectors. That's it from me for today. Do you have any questions?